So those methods are highly used in medical and close uh, types of industry and laboratory practice methods to kill bacteria. But of course, we should understand that bacteria around us is not just present, is not just alive. They have very complex relationships, and all microorganisms around us are interacting one with each other. And of course, sometimes even other bacteria or other microorganisms can be harmful and can kill pathogenic bacteria. In contrast, some pathogens can kill normal bacteria. And of course, we should remember that it is a complex world of microbiology, complex world of microbial interactions, and we need to say a few words about how one bacteria can act on other bacteria. So all types of microbial behavior are divided or combined in two uh, important groups. One is a synergy. Synergy, it is when relationships between microorganisms are beneficial for those microbes. And we have several types of synergetic relationships. First one is symbiosis. As you can see on the picture, symbiosis, we will discuss relationships, behavior of two microbes to make it easier. And symbiosis, it is association of microorganisms when every gets some benefits one from each other. Means that both microbes producing some suitable substances or provide some uh, surrounding beneficial one for each other, but both microbes are in benefit due to that type of behavior. Second type, it is a metabiosis. Metabiosis is this association of microorganisms when one microbe is producing substances used by other microbes as the source of energy. Usually it refers to some bacteria which can utilize, for example, sugars only till acid, for example, and other microbes can use that acid for further release of energy, or some other substances which can be used by one microbe as the source of energy, but it is a waste product for another microorganism. So metabiosis, it is association of microorganisms when one is producing substances used by other as the source of energy. Third type of behavior is a catalysm. Catalysm, it is a type of behavior when one microorganism is secreting factors which are stimulating growth of other microorganisms. It can be vitamins, some other compounds, but it is a growth factor or stimulating factor produced by one microorganism and influencing on other microorganisms. So catalysm, when one microbe is secreting factors which are stimulating growth of other microorganisms. Commensalism, it is a type of behavior when one microorganism is feeding on other microorganisms but without any harm. Means first microbe have sufficient amount of nutrients of different compounds, and other one is taking only small part of them. And uh, if you talk, for example, not about microbes in general, of course, all those relationships can be applied, easily applied on macro world. And if you talk about, for example, microflora are commensal of humans and of animals, because actually flora is feeding on us, but we're not suffering from that. We have enough amount of nutrients, and we cannot use all nutrients. Some are not suitable for us, but microbes can easily feed on us and use those ingredients for their own life. So commensalism, it is a behavior when one microorganism is feeding on other without any harm. And the last type of relationship, it is mutualism. Mutualism, it is association of microorganisms which is obligate for surveillance of both microbes. Means one will not exist without other microbe, and only association of two microbes is essential for surveillance of both microorganisms. So those relationships are in a group of synergy when one microbe is helping other microorganisms. Of course, not all types of behavior in micro world are synergetic, and we have another type of behavior which is known as antagonism. Antagonism, as the type of relationship, can be by several reasons. First, antagonisms can be observed when both microorganisms needs the same nutrient substances and they are present in insufficient amount. And microbes will fight for food, and one microorganism which is growing faster, which is more active, usually that microbe will survive and it will suppress another microorganism. It will grow in lack of nutrients, and it will grow much slower. It will be inhibited and can, as a result, even die. Another reason of antagonism, it is a production of different inhibiting substances such as antibiotics and bacteriocides, which can suppress the growth of another microorganism. In further slides, we will talk about antibiotics and bacteriocides, but you should remember those two names because those substances are very unique and have a very important role in medical practice as well. So, of course, both antibiotics and bacteriocides will suppress growth of another microorganism. It will grow much slower, or it will even die if antibiotics, for example, are bactericidal. The next type of antagonism, it is a production of different wastes which are lethal for other microorganisms. For example, microbe can release different acids, alcohols, and other inhibiting factors which are waste products for first microbe, but they are very toxic and even lethal for other microorganisms. And again, some, for example, milk bacteria can produce a lot of acids which will suppress growth of concomitant flora, and other bacteria cannot survive in low acidity, they will die. So one unique type of antagonistic relationship, it is predatoriness. Predatoriness, it is complete destruction of one microorganism by other microorganism. Of course, it is one of the most radical methods when one microorganism is just killing other organism. It can eat microorganisms directly or destroyed by other factors. And parasitism, again, it is one of the most known types of relationship between, for example, human and worms. We have a lot of different parasites in the form of insects. Parasitism, it is behavior when one microbe is invading other microbe and feeding on it. And in microbiology, again, it is possible when different types of microorganisms are feeding on other microbes. And of course, one microorganism will prevail and another one will be suppressed and will have more poor conditions of its growth. So in different types of microbiological industry and even in medical practice, of course, some types of behavior are used to obtain required substances, to obtain different products. And of course, mainly in medical practice, as a doctors, we will care about how to use those behaviors and different products of that behavior to fight with infection, to prevent infection, and how to use ability of one microbe to kill another microbe. And here, we need to talk, of course, first of all about bacteriocides and antibiotics. So two very important substances which are produced by microorganisms and have similar effect but have different structure. So bacteriocin, uh, it is a proteinaceous toxin produced by bacteria able to inhibit the growth of similar or closely related bacterial strains. Means bacteriocins, they're active against very similar bacteria. And again, we said, and actually, one of the most first bacteriocins described was a bacteriocin produced by Escherichia coli, and it is called colicin. And actually, they usually named by bacteria producing that bacteriocin. And colicins produced by our, for example, intestinal E. coli, they are harmless for strain which is producing that colicin. But if any other E. coli will try to invade, for example, some enteropathogenic E. coli, then those bacteriocins will specifically inhibit growth of that E. coli. But usually bacteriocins not active against other distant bacteria. And some lactobacillus, bifidobacterium, which are present in our intestine, they will not be affected by bacteriocins. So bacteriocins are active only against closely related bacteria. And the major problem with bacteriocins, why they are not used in medical practice, they are proteins. And proteins are very difficult to store. They are susceptible to environmental factors. They have short duration time period. They are destroyed by natural reasons. And of course, bacteriocins are not used in medical practice because it's very difficult to deliver them to the site of infection. And second reason, they're very restricted by activity. And of course, to use bacteriocins, you should know precise pathogen which is causing your disease. Because if you take bacteriocin, incorrect bacteriocin, some, for example, produced by E. coli, but your infection is caused by Staphylococcus. Of course, that colicin will not suppress Staphylococcus. And use of bacteriocins is complex by that reason. Second type of substances is known as antibiotics. Antibiotic, it is a non-protein, low molecular weight substance produced by or derived from one microorganism able to kill or inhibit the growth of other microorganisms. And the major idea, antibiotic is not protein. It is usually low molecular weight, means it is very stable in environment and it is easy to store and easy to use. So antibiotics are non-protein, low molecular weight substances produced by or derived from one microorganism able to kill or inhibit the growth of other microorganisms. And antibiotics are one of the most important, the most complex, and the most numbered group of drugs in nowadays. Of course, we'll talk about them in the separate part of the lecture. And usually, antibiotics are produced by soil flora. They're produced by fungi or bacteria which is obtained in the soil. And soil, it is the place of, it is a warfield for bacteria. A lot of nutrients are here, but again, a lot of microbes are here as well. And one microorganism need to have some weapon to resist other microorganisms. And antibiotic is that weapon. It is named antibiotic because of anti-bios against life. And actually, antibiotics are very toxic substances. They can kill a wide range of bacteria. Spectrum of activity is much higher, much wider. We'll talk about it a little later. But both substances initially produced by bacteria, and they help bacteria to resist surrounding bacteria. They are used for fighting some area. They're produced by different types of microbes to survive an environment. So antibiotics, again, I hope you all know, and that name nowadays is very, very, very popular. Alexander Fleming in 19 1929, uh, he, as it said, he discovered uh, antibiotics. And again, that history is very, very, very interesting and long. Even some books are written about that important uh, fact. And if you're interested, you can read. And again, it is really interesting how it was described. But again, we have some doubts and discussions about date of discovery. But officially, 1929 is the year when antibiotics were discovered. Again, it is a part of our history. And initially, when antibiotics were discovered, it was a very serious and strong hope that humanity is saved of bacterial infections, because finally we found a cure against bacterial diseases. And it is important to know that before antibiotics were discovered, we call it pre-antibiotic era. Uh, a lot of people died because of simple and primitive infections, because of inflamed cut after some shaving, for example. Uh, Almost 25% of women died after natural birth delivery because of infection of genital tract. And of course, almost every dental procedure was invasive dental procedure was complicated by infection. And millions of people died because of primitive infections, primitive nowadays. And without antibiotics, it was not possible to cure disease with some known drugs. Of course, we had a lot of uh, traditional medical drugs, some herbs and other, but it was cure kind of 50-50. Maybe you will survive, maybe you will die. But of course, when antibiotics were discovered, they became extremely important, and nowadays it is the most numerous group of drugs, and we have separate specialty clinical pharmacologists who deal with antibiotics. And if you know, in our university in Smolensk, we have Institute of Antimicrobial Chemotherapy. It is one of the biggest institutes in Europe. It deals with multiple aspects of antimicrobial resistance, of activity of drugs, with helping different companies in study of newly discovered drugs, and perform a lot of different studies and monitoring of antimicrobial resistance in Russia and even some surrounding countries. And we can talk a lot about antibiotics, but again, our time is limited, and we will talk only about key features, key things of antibiotics, and the major information should be provided for you in some clinical pharmacology department or in other departments and further courses. So, because antibiotics are very numerous group of drugs, of course, we have multiple classifications, and antibiotic can be classified according to multiple properties. So, first of all, according to source, we say that antibiotics are initially produced by one microorganism, and they are effective against another microorganism. And one of the first antibiotics discovered, and actually the most numerous group of antibiotic producers, it is a fungus or molds. Some penicillin, for example, the first antibiotic is produced by fungi Penicillium, by species of Penicillium. Cephalosporins are produced by Cephalosporium, and a lot of different fungus can release antibiotics. So actinomycetes, it is again second producer of antibiotics, and streptomycin, for example, is produced by Streptomyces species. Some antibiotics can be produced even by bacteria, and we know that Bacillus subtilis is producing bacitracin used in medical practice in intestinal surgery, for example. Even some animals can release antibiotics, and lysozyme is one of the most known enzymes in our cell. And saliva, for example, and is in highest amount it is identified in chicken eggs. It is a substance which have natural antimicrobial activity and it is produced by animals. And of course, you all know phytoncides, usually garlic, for example. Garlic is used as example. It is a substance which is traditionally used in multiple countries to prevent viral infections, to prevent bacterial infections, because really substances present in plants can have antimicrobial effect. According to target, we should know, and later we'll talk about different modes of action. All antimicrobials are classified in multiple groups according to which type of microorganism they can affect. And we said that we can have four main type of microbes in human medicine. It is bacteria, virus, protozoa, and fungi. And of course, all those microbes, they have absolutely different structure. And we have different drugs against every class of microorganisms. We have antibacterial antimicrobials. We have antiviral antimicrobials. We have antiprotozoal. We have antifungal. And some authors, some doctors even say that anti-tumor drugs, they as well belong to antibiotics because they're able to kill alive cells. And anti-tumors are fifth class or group of antibiotics according to target. And of course, we should remember that antibiotics in classic understanding refers to antibacterials. And antibacterials should be used only against bacteria. If you have viral infection, antibiotics will not work. You need to use antivirals, and it is a common error in multiple specialties of medical profession. Doctors prescribe antibiotics when subjects suffer from viral infection. They will not work. And in contrast, if subjects have some, for example, protozoa, it's not reasonable to prescribe antifungal drugs or some other drugs because we have unique group of drugs targeted against protozoa. And of course, bacterial infections should not be treated with antiprotozoal drugs. Next classification is according to origin. Initially, all antibiotics were derived from microorganisms, and nowadays the group is called natural antibiotics. They are produced by live organisms. But of course, natural antibiotics have multiple penalties. They are less stable in environment. They have multiple side effects. Activity is very low. And after some time period when penicillin was discovered and natural antibiotics were identified, people tried to modify, to change something in structure to make those antibiotics more stable, to make them more active. And those natural drugs modified by human are known as a semi-synthetic. And for example, amoxicillin is a semi-synthetic drug that is initially modified penicillin. And of course, nowadays we have synthetic or artificial antibiotics which are completely designed and manufactured by human, and structure of that antibiotic is not present in environment and natural life of our planet. And fluoroquinolones is the most famous group of drugs which are completely designed in a computer and they manufactured by human only. So one more problem of antibiotics still, they have activity against only known microorganisms, and ability of antibiotic to act on some bacteria is known as a spectrum of activity. And according to spectrum of activity, we have three group of drugs: wide or broad spectrum. It is an antibiotic active against both gram negatives and gram positives. And for example, fluoroquinolones are active against both negatives and positives. And again, we need to remember that structure of bacteria is different. And we have some drugs which are active only against gram negatives or only against gram positives. And of course, if you're not sure in your diagnosis, when initially you see that infection is present, but you have no idea, absolutely no idea, which bacteria is causing the disease. For example, it is a wound infection. It can be caused by E. coli, it can be caused by Staphylococcus, by gram-positive, by gram-negative. You're not sure which bacteria is here. Of course, it is reasonable to prescribe broad-spectrum antibiotics. But when you took a look in the microscope and you see that only, for example, gram-negatives are present in your sample, then you can switch to narrow-spectrum antibiotics. And for example, polymyxins, are active only against gram-negatives. They are absolutely useless if infection is caused by gram-positives, but they are more active against gram-negatives than some broad-spectrum antibiotics. And of course, if you know that your infection is caused by gram-positive or gram-negative, it's better to prescribe narrow-spectrum antibiotic. The main idea that antibiotic always need a target. It needs something to kill. It is a substance which have only one purpose of existence. It is kill something alive. And if you have a pathogen, that antibiotic will kill the pathogen. But if pathogen is not present, that antibiotic will still look for a target. And in that case, it will find normal flora bacteria. And antibiotics will destroy bacteria which is normally present in intestine. And of course, in that aspect, it is more reasonable to use narrow spectrum activity because antibiotics will not affect part of the normal flora. They are more safe for normal flora bacteria. And we have very few drugs which have limited or targeted spectrum. Limited or targeted spectrum refers to a drug which have only one indication of use or activity against one of few bacteria. For example, clindamycin is the drug with the targeted spectrum or limited spectrum of activity. And again, those antibiotics should be prescribed only when you're sure with the diagnosis you have identification till species of microorganism. Uh, According to effect, we say that antibiotic is a substance which can suppress the growth of bacteria or kill the bacteria. And similar to antiseptics, antibiotics can be bacteriostatic or bactericidal. Bacteriostatic antibiotic, it is antibiotic which can inhibit growth and multiplication of bacteria. For example, chloramphenicol is a static drug. And we have bactericidal drugs. For example, penicillin is bactericidal. It can kill microorganisms immediately. And sometimes we have combined effect. Combined effect, it is a phenomenon when antibiotic can have different effect when prescribed in different concentrations or they are prescribed in different combinations. For example, macrolides, they're bacteriostatic if you prescribe them in low concentration. But if you give it in high amount, they will become bactericidal in effect. And we have, for example, cotrimexazole or trimethoprim sulfamethoxazole. In mono formula, if you will prescribe monocomponent, for example, trimethoprim, it is bacteriostatic. But if you combine and sulfamethoxazole as well is bacteriostatic when you will prescribe it as a monodrug. But if you will give it in combination, cotrimexazole, the drug have bactericidal activity. And it is a phenomenon when antibiotics have combined effect. Rare, but it is important to know because we can adjust our therapy by use of different concentrations or different combinations of drugs. So, how antibiotic can kill bacteria? What that chemical can do to destroy a live, highly active, multiplying microorganism? So we have five main modes of action, and all antibiotics are classified into five groups according to their mode of action. So penicillin, the very first drug, is a drug which can damage peptidoglycan. It causes damage of peptidoglycan. and inhibiting synthesis of the cell wall. Without cell wall, bacteria, of course, cannot exist. We talk about protoplasts and spheroplasts. We know that we have some exceptions. But cell wall synthesis inhibitors are the first group of drugs, antibiotics discovered and described, and mode of activity is very good studied and prevalent in multiple drugs. So, inhibition of cell membrane. We know that membrane is required to perform different processes of uh, absorption, exchange of different energy, minerals, water with environment, and of course, if cell membrane is affected, uh, multiple processes will be inhibited, suppressed, or even blocked, and bacteria cannot survive in that state. And different drugs can have different effects. Some can affect structure of cell membrane, some can affect functions of cell membrane. And again, we already mentioned polymyxins. Polymyxins are drugs with narrow spectrum of activity, and they have mode of cell membrane inhibition. Third group of drugs can inhibit protein synthesis. Again, aminoglycosides, for example, acting on synthesis of protein. And different drugs can inhibit translation of proteins. They can affect proteins, so they can block, for example, ribosomes, and synthesis of proteins will be blocked. One more mechanism of activity is nucleic acid inhibitors. Again, fluoroquinolones, synthetic drugs, are nucleic acid inhibitors. They can inhibit synthesis or block nucleic acid itself to prevent reading of information. And of course, we have DNA in bacteria is the main type of nucleic acid, and fluoroquinolones blocking functioning of DNA. And the last group, it is a group of competitive inhibitors. It is a group of drugs which are very similar in structure with some essential growth factors or vitamins, but they can perform no function. They are looking like vitamin. Bacteria will take it, it will try to use, but that antibiotic will block enzyme, it will block some important pathway. And anti-TB drug, isoniazid, for example, is a drug of competitive inhibitor group. And of course, knowledge of mode of action is important, especially if you need to perform combination therapy. If one antibiotic is not sufficient in your therapy, you need to combine several. Of course, it's better to use antibiotics with different modes target, and one antibiotic will suppress another, it will block effect of another. Of course, it's much more correct to combine drugs with different mechanisms. One, for example, will block cell wall synthesis, another will block cell membrane or some ribosomes. And effect in that case will be much more beneficial. So according to chemical structure, we have multiple groups. I will not even list all of them. One of the most prevalent group is the beta-lactams. It includes penicillins, cephalosporins, carbapenems, and some other. But actually, we have multiple different chemical classes of antibiotics. Again, it is important to know by several reasons. First of all, if you need to combine different antibiotics, it's better to choose antibiotics with different chemical structure, because first, resistance to antibiotics, we'll talk later, efficacy of antibiotics in one group can be very similar. If macrolides are not working in some current infection, it is a high chance that no macrolide will be effective. Second reason, if your subject have allergy on some drug, usually that allergy is common for the whole chemical class. And if your subject have allergy for one macrolide, usually all other macrolides will be the same allergic. And if you know that your subject have or had hypersensitivity to some drug in the history, it's better not to use any drug with similar chemical structure. So, of course, we listed all important classifications of antibiotics, but of course, it is important to know the property of every antibiotic according to every classification, because again, usually allergic reaction is common for class of antibiotic, for chemical class, or if antibiotics are produced by the same microorganism and subject have allergy on microorganism itself, then any drug produced by that organism will have common or cross reactivity. And in that picture, it is a penicillium fungus showed growing on culture medium and electron photo of the fungi. And classic benzyl penicillin, for example, according to complete classification, it is a natural beta-lactam bactericidal by disruption of cell wall synthesis antibacterial drug with wide spectrum of activity derived from penicillium. Complex, yes. Difficult, yes. But every point is important if you need to prescribe antibiotic to your subject. And it is the reason that complexity of classification is the reason why we have separate specialty of doctors, clinical pharmacologists who deal with antibiotics, because it is difficult to remember every drug and all properties of every drug. And of course, use of antibiotics is a complex uh, and difficult thing. And if you're not sure with some prescription, it's better to consult to specialists, to call clinical pharmacologists and ask for advice in appropriate person. And of course, antibiotics are a very complex group of drugs, and you should study it very good if you're going to work in infectious department and surgery or in some other hospital settings where bacterial infections are present.